We're going to move into the first of two focused discussion topics that we're going to try to tie together some of the things we've been talking about so far. The first focused discussion is on what can NHGRI do to facilitate bridging molecular and organismal phenotyping. We've been talking about, we're not phenotyping, but phenotype. We've been talking about this a bit already, and the idea would be how to make better linkages between molecular and organismal phenotype, and are there things in between that we need to be considering? So I'm going to turn this over to Andrea.
So thanks for enduring. I think this is going to be a little bit of a different type of session, and the goal is not that of sort of presenting state-of-the-art in some areas and then asking questions or to summarize a group meeting, but really more to challenge the audience and with potential where we're going to go next, where NHGRI is going to go next. And I want to say that I'm only selected here as the spokesperson for this group because I have absolutely no interest either in terms of
financial interest.
Either in terms of being funded or seeking funding from NHGRI in the foreseeable future. So I can be as obnoxious as required to make some points. All right, so the goal of this session, we actually renamed it a little bit to focus somewhat more because it ended up being a little bit vague in the context of the previous ones, is essentially, can we go beyond cataloging and still remain faithful to the original NHGRI mission? And basically, can we use these approaches to go beyond sort of associational approaches to sort of go between genotype and phenotype, and introduce a layer of mechanistic understanding. And so, and obviously, my co-chair, Barbara Wold, and Len Pennacchio have had major input in putting these slides together. So, so the first thing that was obvious is that even though we have done a lot, we have characterized maybe 1% of the regulatory genome, et cetera, there's still a lot of genomic information, and do we need a new class of sort of genomic class phenotyping, including transcriptomes, epigenomes, DNA accessibility and structure, DNA RNA modification. And I think one question that came up pretty frequently across the various discussion groups is, do we need a complete catalog, or can we actually work off more specific one, more specialized one? That is, do we need to know everything in order to model and understand something? The other area where I think there was really broad consensus is that in order to really understand how the genome is regulated, we probably need to go sort of outside of the comfort zone of the genome and understand how also the genome regulates, which is not necessarily through its own intrinsic regulation. And so do we need to characterize other modalities such as proteomics, metabolomics, high-resolution molecular imaging, and you don't have to read, you can read all the rest, but also we have to recognize that the some of the phenotype actually really change under perturbation. I think Botstein did a very famous experiment showing that unless you perturb the cell, you don't see the relationship between certain gene and the phenotype. And also, is there an opportunity for other metadata approaches, that is, approaches that develop technologies, analytical tools and methodologies, and importantly, modeling? For instance, should we extend the ENCODE program to encompass post-transcriptional, post-translational, and single transduction regulation? And so I think the question is here also, is the goal to build universal catalogs or to use these catalogs to build more predictive, specific, and interpretable models? The third point was the sort of understanding this kind of layered gradation of compartments in which these which genomes play a role, going from better understanding of what we call endophenotypes, that is molecular level phenotypes within the cells, to cellular level phenotypes, which are more like exophenotypes. I think Michael made that point before. Going to cell-cell interactions, and which are also, they're studied quite extensively. I mean, the Nobel Prize this year was awarded for cell-cell interactions, but there's no systematic way to do it. In fact, I was really shocked by seeing that in PubMed there were no papers providing sort of systematic approaches to elucidate cell-cell interaction. And then finally, organismal phenotype, and do we, and is there essentially a gradation in which one of these layers informs the others? And again, some of the phenotype relationship may only be appreciated under specific perturbational context and within a specific cellular context. The final point that I wanted to make before sort of opening it up is that I think there's a couple of lessons that have come both from human genetics and for studies in cancer and I think this goes back to some of the things that we've heard yesterday. I made the point of this combinatorial complexity of genetics, where even having 1,000 variants contributing to a trait may lead to more than the number of atoms in the universe in terms of patterns that may yield different penetrants. And the question here is actually, do we have to understand each one of those independently, or are there integrative mechanisms within the cells that allow us to simplify and universalize this knowledge? And I think both some of the theories built around the omni-gene hypothesis with core genes and the similar type of theories built in cancer around master regulators suggest that maybe there are mechanisms that are highly integrative in biology that allow us to focus on the relatively small number of distinct phenotypes as opposed to the infinite number of ways in which these phenotypes can be implemented. And there are many specific examples of that. So basically, this is just opening up to the crowd and raising up a few discussion points. You can read them. I don't even need to go through them. But essentially, what phenotypic assays, what are the scale, even temporal scales? Do we need new assay development? Scale up in combinatorics for assays, a point that I think Aviv raised earlier. Building and evolving phenotype priority matrices, what is the times that we need to deal with, and how complex is that going to be? What constitutes a comprehensive matrix? Can we define that in vacuum, or do we need to define it in the context of a particular thing we're trying to understand? And then finally, data mining and integrating across different phenotypic data and modeling. I think that came up across many of the different areas of discussion that we've had. So at this point, maybe we can open it up and sort of hear comments and either profound disagreement or profound agreement or anything in between.
The more specific and pointed a proposition is, the more happily it will be received.
Yeah, and also the more useful towards directing the NHGRI in terms of defining future programs and priorities.
So I could speak up also. I mean, I thought from the last session, this idea of we want to anchor around something, and Howard and others have put forth that half or even more than half of the genes have no known function, and that's a project that will require technology development, variety of types of phenotyping. We talked about the population genetics and human loss of function alleles, that it makes it something that almost any of us can contribute to. And there's brand new biology to be learned in all these genes that have no known function, and it will link back to the clinic. And it's something that, if you wanted to throw out a moonshot saying to determine some level of functional understanding of some fraction of those genes, it is tangible, and so it's an idea to consider.
Yep.
So, one thing that I think needs to be done and defined is when people say genes, that needs to also be further defined. If they're talking about protein coding sequence, non-coding areas, anything that's transcribed into an RNA or regulatory regions or what, because those are different issues to tackle, and they expand further and further when you get away from the actual coding sequence itself.
Good point. I think originally thinking about open reading frames, but I think the same thing can hold true for, is there a gold standard of regulatory elements that are enhancers that we should try to understand how they work? We know very little about the protein-bound mechanisms of how these things are functioning, and you could have a gold standard that is what we all rally around also.
Yeah, but I think also that we should try not to be parochial in terms of sort of highlighting one layer of regulation versus the whole set of mechanistic regulation occurring in the cell. So if you talk to people that work in signal transduction, they will tell you almost everything is interpretable by MAP kinases and other processes that occur post-translationally. If you talk to people that work in transcriptional regulation, then everything is done at the level of regulating gene expression. I think the cell cannot be understood without really going across all these layers. And some of the most recent modeling efforts have actually shown that a lot is to be gained by providing an integrated view of that, and that will inform the genome and will be informed by the genome.
Jay?
Yep.
So, this is Carol Bolt, Jackson Lab. So, under the data mining and integrating across diverse phenotyping data, I think one of the things that needs to be folded into this is ensuring that the data that are generated are annotated so that integration is actually possible. So I mean by this sort of the metadata, the semantic layer that goes on these data so that they're findable, accessible, interoperable, and reusable. And there are a number of standard ontologies out there to use in this regard. There's a mammalian phenotype ontology, the human phenotype ontology, the disease ontology. There's a number of ways that we can facilitate achieving these goals if we all adopt and use these common standards.
Yeah, I think this goes to a really important point, which is that many times before in other part of this meeting, we've had the question of whether something is actually NHGRI specific. Why is this NHGRI specific? Why isn't this done by other institute? And I think what you just pointed out, that is the ability to actually turn even episodic discoveries into something that can generalize and then be used as a framework to understand the entirety of genome regulation. That's what makes something, I think, very NHGRI specific. So I think that's something that we should actually keep in the back of our mind.
Well, and NHGRI has been at the forefront of developing many of these ontologies. Gene ontology is a good example.
Yeah.
And in fact, some of them are being applied in these contexts. So attempt to apply an appropriate ontology, for example, to the ENCODE samples. But just speaking of that one small case, how very difficult it has been to make that sensible to all users, because decisions were made for a host of reasons by some people. And making the data findable is always considered to be, oh, yeah, that's necessary, we'll do that. That's probably one of the most enormous challenges. So
you had a, yep.
This is Jason Benrosha from Harvard. So maybe in a naive way to think about it, and I guess of one direction NHGRI can go, one thing to think about is how many of these phenotypes can be read out in a gene expression level, right? And how many of them can be, which those that couldn't be converted to sequencing-based readout. Of course, now with the advent of single-cell genomics, and this just being the kind of data currency for a lot of biological discoveries, it'd be really nice to have computational tools to pull this out of the data that already exists or will be generated, and perhaps would couple well across different groups where one could say this is the gold standard way of reading out this phenotype, and how far can we get by just sequencing data alone, right?
Yes. Any other comments? Aviv?
The discussion is around bridging the molecular and the organismal phenotyping. And I think one of the greatest challenges around that is pleiotropy, is the fact that the variant could have multiple functions, and we could assess its molecular impact. And if we assessed it too narrowly, we could follow the wrong molecular impact that actually is not related to the organismal phenotype. It's related to some other phenotype that is not the one we started from. And it can take a long time to figure that out. And so I think there are two aspects to this that matter a great deal. The first is being very comprehensive in context, so that we're not assessing just in one context that appears to us to be the correct one up front. And the second is to get as close as possible to the relevant context early on, because this digging thing is substantial, monumental. And finding where is the right place to look at a variant is something that I think is now within reach. But absent it, I would be concerned about a failure mode where you go all out on this and you end up walking down the wrong path. And people saying, well, the whole thing was not useful. It can be useful, but it has to be done right. Yeah,
so I think thank you for raising that point. I think this is a really important issue that also goes beyond that. It raises the further issues of what are the actual models that can be used to really address pleiotropic regulation. Are all models bad? Are some of the models better suited at solving certain problems than others? For instance, we know that we can reprogram a fully differentiated cell to an IPS by just topically expressing 3 factors. So there seems to be simplicity in that context, but then again, there is tremendous complexity of regulation of the specific developmental cascades that arrive. We're going the other way around. And so, yeah, go ahead, Ahid.
I wasn't even going that way. Mm-hmm. So I wasn't going down the, it's a bad model, it's cell culture, it's an organoid, it's the mouse, it's not the human. Nothing like that. Even inside the human, a variant is present in all of the cells of the body. and it will have actions in those cells. And even within the one cell, within one context, it can have more than a single molecular effect. And knowing how to chain that, right? We accept upfront that between the variant and the organismal phenotype, there's a chain of events. And it can fork and branch in different ways and merge and so on. But some of it, because of pleiotropy, it's just going on another direction. It's not related to the phenotype we're interested in. It's related to a different one. But it might have the larger effect size in the molecular realm, or we might be looking at the wrong cell type or the wrong tissue, and so we would walk down that path and we'll never meet the phenotype we're interested in the first place.
Yeah, I wasn't trying to put words in your mouth. I was just saying that what you raised the additional issue of how do we actually validate and even think about these things in Eric.
Yeah, just to underscore it, it underscores again why a comprehensive picture is helpful. If enhancer over here regulates 2 nearby genes, and you get all attached to gene number 1 as the thing that's related to your disease, but it's really gene number 2, and you didn't know, you're in trouble. Same if it's in a different cell type. So I think it really underscores why comprehensive data makes it harder to fool yourself, because you at least know what the other alternative choices are. Very often in chasing down the basis of disease, people are going on the thing they're looking for. And I think that's the problem with pleiotropy.
So, Eric, and Aviv, isn't it also the case that this is an argument that at least a big chunk of this is well placed in model organisms where the variant genetic background is not going to be confused with pleiotropy as you do it? So if you're on a clean background and you're asking how many different cell types and organ systems are affected by Notch, you can manage to do that. But if you're also on a very genetic background, you're going to be you're going to get a richer answer.
Well,
and possibly confusing.
Yeah, yes and no. I mean, yes, of course, model organisms are good, and we ought to force ourselves, too, to figure out where in the human, through isogenic editing in cell types, we can do that, where we have heterozygosity and we can tell allele-specific effects. So I think it mostly says why this is well lodged within NHGRI, because whether it's model organisms or humans, NHGRI is the place that tends to be committed to taking, these kind of comprehensive, rigorous kind of views that are complete enough to know anything. So I agree, but would expand. And
always fine with that. Will you pay?
I thought that was a good compromise. You also mentioned something I wanted to just raise a second, Andrea, which was you said that we wouldn't be able to see effects unless we looked in just the right place, maybe, or under the right circumstances.
Some of them.
And sometimes. I just wanted to throw out that, and this is a point that Viv makes to me, so she could make it, but I'm here. So randomness, random fluctuation within cells can reveal dependencies that we don't see at the higher level. So again, collecting single cell data and examining it not just for its average behavior, but for the fluctuations and harmonics that it teaches us can be extremely valuable. And yet another reason to be very systematic and rigorous about seeing those things. So I think a lot of what's going to happen in the next couple of years is we're going to press ourselves to get more and more rigorous data out of the data sets we collect. Yet another reason why this has to go through NHGRI, because these can't be just byproducts of other studies. They have to be studies designed to have these sensitivities.
Yeah, I think there's a continuum of effects. And so there are things, for instance, if you need to study activated T cells and you have non-activated T cells, it's going to be hard to rely on fluctuations, right? But there are certain things for which fluctuation have been incredibly important in modeling. The same thing, I think that the dialogue here is not whether we need comprehensive assays to measure everything in the genome, or we need things that actually specialize. I think we need both, but the problem is breadth versus depth. So I think in a context of doing genome-wide everything, we will achieve a certain level of understanding that can then be specialized to really understand some phenotypes in extreme detail and mechanistic detail. So I think that is the sort of boundary between the two. I had a slide here that I prepared, which was this basically, where you can see that, I don't know, where you can see that continuum. So basically saying we can do associative learning by mapping no mechanism. We can do an unrealistic mapping of every single detailed mechanism. But there's somewhere in between where we can actually learn something important by doing only 20% of the work that would be required to do it in everything. Other comments? Yeah.
Yeah, I'm Laura Mamunis from the Neurology Institute. And, there's been a lot of discussion about phenotyping, and of course a lot of it's at the molecular cellular level. But I guess the question is, does NHGRI want to wade at all into really complex behavioral, complex neurological phenotypes? And I say that because, I'm involved in the UDN with Hugo, and, about 50% of the phenotypes that we're seeing are complex neurological phenotypes. And, we're asking our model organism screening center to try to model those. And, they've actually had some success in fly and worms. And these organisms, don't have the evolutionary complexity that humans do, for example, to really, understand language or social communication or anxiety or you name it. So, are there ways that NHGRI could help us, like I'm at the Neurology Institute, to, better use model organisms to characterize some of these very, very difficult phenotypes that even a mouse model is not the best way to study these? You may have to go to a marmoset, but that's so low throughput. So that's my question.
So Tom, do you want to expand on that? Because you raised a very important point, for instance, about modeling some of the variants that are known to have extremely high penetrance in ALS in a mouse, and only one actually produces effects that are recapitulating what happens in the human context.
Yes, I made a point earlier that ALS serves as an interesting example and challenge to this approach. That obviously the best system so far is the mouse in principle, but about 30 genes have been identified that are either dominant familial or sporadic mutations, and that except for the overexpression of SOD1, there is no mouse model that really reflects the disease in any convincing way. This is true for TDP43, it's true for C9orf, and there are little hints that they give you some fraction of an insight. And so that's obviously a big problem. You can't do knock-ins in mice with ALS mutants that are clearly, very robust and dominant mutations in C phenotypes. So it's at least in ALS, it's a real challenge, and I think you could argue that in Alzheimer's and Parkinson's, similar issues are there.
And it's the same with epilepsy and a whole bunch of other disorders.
So if you want a real great challenge for NHGRI that I don't think is being addressed by any other institute, is to create quantitative metrics to assess how relevant a particular model is to studying a particular phenotype. I think this is something, for instance, in cancer we rely on histology sometimes in the cell. We rely on a few markers. If you actually do a comprehensive analysis, you realize that only about 10% of the cell lines that are available in the CCLE are actually truly modeling the major characteristics of the corresponding tumors, and the rest is just some bizarre organism that doesn't exist anywhere in a human. So I think being able to do that for the mouse, for the fly, for cell lines, for organoids, even for primary cell passage in short time, because they change quite rapidly. This is something that could be a grand challenge that only the NHGRI is probably set to address.
So I mean, just to follow up, Hugo had said earlier something very intriguing, that he could use the wingless phenotype in the fly to understand cancer. So, there's a perfect example of understanding that gene function in the context of that organism could that be then informative for other organisms and how that gene functions? But anyway, I'll leave it there.
Yeah, I think that Rodney Rosling would argue that everything we'll learn about DNA damage response is from learning yeast. So I'm saying that those are things that are very important and can be studied in model organisms very effectively. So not everything requires the full complexity of the human cell. Yes.
If I can try to reduce at least a subset of the discussion to a specific proposal. So you could, and I was talking a little bit to Barbara about this at the break, but you could conceptualize this as, what are all the things that we would like to knock out, let's say, and what are all the conditions we would like to do it in? And that in itself is a giant matrix, which is, ideally we would get the entire matrix, but it's probably unrealistic. But you could imagine trying to be comprehensive, as Eric was saying, both in, within individual rows or individual columns. So picking, for example, a subset of conditions, let's say, even encode cell lines supplemented by some other things, but a modest set of conditions, and trying to knock out every gene and every functional element and to measure in a limited number of molecular imaging assays, or, a selected number of things, the functional consequences of all of those knockouts in a quote global way for the rows. And then for the columns, you pick a subset of genes and or elements, a small number, and you go as deep as you can in terms of context exhaustively across model organisms, cell types, et cetera, to try and actually put bounds on what the complexity is there, which we really don't know. But that's one way of thinking about it. It's two different but related projects that try and attack this while still being real.
Yeah, no, I agree with that. To a large extent, I think another way of thinking or talking about exactly the same problem is, what is the minimum fraction of the genome that we need to, and the genome and the epigenome that we need to fully understand in order to be able to study a particular problem? And is there a generalizable, universal way of taking a particular problem that we want to study and finding out what is its molecular implementation to the level where we actually can generate those matrices without doing it 100%, maybe only with 10% or 5%. I think that would be really valuable, and we already know so that if you look at specific gene variants, or maybe a different way of thinking about this, instead of connecting the phenotype to the genotype, is connecting the genotype to the phenotype. That is, starting with genes that we actually know have had some major role in particular disease, and building all the machinery that surrounds them, independent of whether it's transcriptional or not, that allows essentially to implement their phenotypes through effectors, other modulators, co-modulators, et cetera. Yep. Mike.
So I think this is a little bit of an idea that I forget either Eric or Richard brought up in the previous session, which is it would be nice in the process of doing that to have some limited set of cellular phenotypes that are independent of the actual function of the protein, whether that's transcript quantification, protein folding, protein modification, whatever, but something that would tell you doing this to this cell changed something, whether I know what that thing does or not, but just I did something, did it matter in this cell type? Because just knowing that it didn't, comprehensively to say I put this change in this cell type and that didn't do anything. I don't need to look at this cell for this change anymore. I think that would be very useful, but I feel like even with everything we have from ENCODE, we have this enormous depth of data, but we don't necessarily know which of those things are necessary and sufficient to tell us that something changed in the cell.
Yeah, I think we're saying exactly the same thing. I agree completely. Yes?
I guess I'd hate to see this experiment design where the only perturbation was knocking out each gene, just because, I mean, think of all the examples of human diseases where different alleles are totally different phenotypes. We heard the example of ACOX1 that Hugo gave earlier. We'd miss an awful lot of biology if that was the only thing.
All of haploinsufficiency would be lost that way. So I think that this is just an example in terms of one of the modalities that we can use, but I think when we talk about perturbation, we need to think about drug perturbation, environmental perturbation. And I think to some extent, one thing that has been really helpful in other contexts is to use genetics as a way to understand what are the integrative mechanisms that then also integrate signals from the environment and from other cell-cell communication. For instance, in breast cancer, luminal subtype, you have not only specific germline variants like FGFR2 that are really penetrant in a disease, but you also have environmental factor like exposure to, long-term exposure to estrogen that actually impinge on exactly the same molecular mechanism. So one really helps the other. So this is something that could really address some of the points you make. Yes?
So rather than just simply deleting the gene or whatever, what about instead just doing CRISPR activation or CRISPR inactivation? And you could even get to greater granularity of like just inactivating a particular promoter if there's more than one promoter that particular.
I mean, now there's an incredible number of techniques. You can bring degrons to proteins to degrade the protein directly. You can modulate RNA stability. So I think that the technologies that are now being available for doing that kind of sort of manipulation of the genomic, the gene products is almost infinite. I don't think we need to sort of address the specific ones as much as opening up, I think, the entire repertoire of things that could be done if such an approach were used effectively across the board. Yes.
To pick on Andy's point, and you're Andrea, in addition to perturbations that we design, and in addition to just the fluctuations in biological systems, which are a form of perturbation too, there is human natural genetic variation, and it's a great thing, and we should use it. Which means whenever we think about these sophisticated readouts in systems that we engineer, we should also think about human studies where we can apply the same measurements, but we actually get natural genetic variation acting for us, and ideally in a human body and not just on a dish. But even with just diverse cells in a dish, it's so doable now with such high resolution. I think we shouldn't lose sight of that, especially in the genome context. And it can be done in a somewhat disease agnostic way, because this is common genetic variation. So at least for the basic mechanisms, we can go quite far without being attached to a very specific system, which protects us a little bit from this pleiotrope problem, and at the same time is not something that one of the other ICs would do.
Yeah, I don't think you're picking on me, because this is completely espoused by what we do, an entire community does. That is, it's actually been shown that using drug perturbation to reconstruct regulatory mechanisms is much less effective than using natural occurring variability across different patients, for instance, in cancer or in other disease studies. So 100% in agreement. Yeah.
The possibility to pair something like organoids where you take the entire genome and are looking at whatever that genome is versus a controlled setting in which you CRISPR-Cas9 do these molecular things for particular mutations that correspond to source of origin and have that paired setting could be quite pretty. Yeah.
I just wanted to echo what Aviv was saying. I fully agree. I just want to sort of highlight the fact that if we want to do this at scale and use naturally occurring variation in human populations to understand molecular mechanisms underlying GWAS associations and whatnot, we need large sample sizes also for the molecular measurements. We can map cis-EQTLs with a couple of hundred samples, and that's a good start. But we are also, after doing this for years and years, we are starting to see the limitations of that, and also the promise that there is in larger sample sizes. Luda Franke has now mapped trans-EQTLs quite efficiently in blood samples of 30,000 individuals in a meta-analysis setting. And you are actually starting to get really, really valuable insights into regulatory pathways and sort of downstream effects of genetic perturbations in an in vivo or ex vivo setting. But you really need thousands, if not tens of thousands of samples with molecular data to be able to do that. But I think it would actually be worth it. And it's something that is kind of sort of study design, big resource type of a project that I think few institutes would do, if not NHGRI.
Yeah, so even as of today, I think that people require large cohorts, but if what is being proposed here, essentially bridging phenotype to genotype using intermediate molecular mechanisms, is truly implemented, I think we should be able to do, and this could be a grand challenge for the next 10 years, we should be able to do genetics on the single individual or even the single cell. Because essentially, if you build a model that is informative enough, then you no longer need to do statistics. You can simply trace back in a causal mechanistic way. And I think this is what biologists have been doing, one little piece at a time, when they elucidate some of the major element that controls cellular behavior in isolation. So I think absolutely, at this time, it is absolutely required. But I think the grand challenge would be, how do we move beyond statistics and how we do beyond GWAS, and we go into really exploiting quasi-mechanistic genome-wide regulation to interpret the role of genotype.
So natural variation is my bread and butter, so it's hard for me to argue against it. But the design, if we only looked at natural variation in humans and required looking at 10,000 at a time and saw the effects as just the average over those 10,000, we're looking at the marginal effects collapsing over all those other variables. And in a situation like that, we would get what we get with GWAS. We would see no interactions, we wouldn't infer networks. All the other signal would be averaged out and smeared away. And so I think we do need a combination of natural variation to identify interesting variation, but also perturbations to have a controlled background to be able to make sense of it.
Absolutely. Yeah, Viv.
Just in response. Absolutely.
Let me ask Tim.
Sorry.
Yeah, I was just going to say, you're always going to need statistics. You're not going to be able to move beyond that.
I'm sorry?
I was going to say, you're always going to need statistics. You're not going to be able to move beyond that. You said moving beyond statistics. We're always going to need it.
We're always going to need statistics.
Always.
I don't think so. I mean, I think that you can do, people were doing statistics where they were trying to understand the law of motions before Newton came up with the three laws. After that, you don't need to do. You just build the Hamiltonian, and it's completely deterministic now. So I think if we can build a similar model, we won't probably maybe never get there, but we can get relatively close. Maybe we can avoid having to do statistics. Aviv.
Just to say that I totally agree with Andy and others that you need both. I was just trying to say it's also okay to use human genetic variation. Yeah. The second is that I think with certain higher resolution approaches, all sorts of opportunities open up. For example, the ability to do allele-specific expression is higher when you have the information at a single cell level, and you can use multiple sources of variation in the data set simultaneously. So not just the genetically driven ones, but also ones driven, for example, by these fluctuations. We will have to be creative if we want to solve problems of this magnitude.
Elise?
Yes, I just want to point out that we've got 5 minutes left for this discussion, and love to hear from some people who haven't yet spoken up, or and if there are some ideas that haven't been aired yet.
People who haven't spoken up, please raise your hand.
All right. This is Andrea Rady from Oregon Health and Science University. This is something that it's been brought up a few times already, but I think it's important is the thing about imaging, right? It's a great way to look at other phenotypes that's not just at the organism level, but cellular architecture within a structured tissue, things like that. It's really quantitative. You can get a lot out of it, and tying that to genomics and epigenomics. So I just think that's an important area to think about.
Yeah, molecular imaging was absolutely captures complete agreement. Anyone else?
Just putting a...
You already spoke, so you can't speak again.
Can I just put a coda on what he said? The thing that's nearly right here, almost ready for prime time, but will definitely be in the next 5 years, is the spatial MERFISH, seqfish kind of RNA. And that puts you right at the imaging level data and the shapes and sizes and neighborhoods of cells. And that's in graduating from the straight molecular to the molecular plus cellular level, and even in helping to, single cell, as it's currently done by disaggregation, is an exercise in insight by disaggregation, and then we're going to have to put it back together. And that may be one of the linchpins that is time ready for this NHGRI period.
So Steve?
Yeah, I wanted to follow up on that because I think the next 5 years we will have these technologies and we can begin to address what I think is one of the hardest questions in genetics, and that is time. The effect of a genetic variant over a period of time is not a continuous process. And so the model organisms are a terrific place. In cancer, turning out to be very helpful with the organoids. At certain steps, you generate one mutation, and then you bring a second one in, and you start to see very different things that you can model and or get mechanistic insights. So I think if we're thinking about this, whatever we do, I think one of the standards that seems to get lost in this is really what's the temporal nature of these things. Is, you know, having that germline mutation from the start is one thing, but introducing it, what does that mean? What has been lost, so to speak, in understanding the developmental process that would shed light on those mechanisms? So I think trying to bring together this data and have standards to be able to help us to interpret that, because I think many people often misinterpret models because they haven't necessarily taken those things into account, and we certainly see that in spades with cancer and all the mice and zebrafish that we can cure when we have, you know, conditional knock-ins and knockouts. It's very different from what happens in humans per se, and I think this is something we have to really be thinking about, and it's a place where many, many different diseases will collide over this, and NHGRI could be very helpful in figuring out how to address some of these really fundamental questions.
Yep, noted completely.
Yay, dynamics. I think they're telling us we're out of time
We have
course.
2 minutes and 12 seconds.
Oh,
so,
okay.
Yes.
I think one thing, we've focused a lot on looking at how sequence variation implicated in disease leads to a phenotype and how we can use these molecular technologies we've got to look at this. And then we get into very detailed discussion about what is the phenotype. But I think another way to look at this, and a focus of NHGRI, is to understand how the genome works. And 90% of sequence variation is within non-coding regions, putative enhancers, and people say enhancers are too hard to understand. But I think that using these technologies just to understand how do you break things is really important. And that way you don't need to know per se exactly what precise phenotype it has. So for example, the lady asked, well, I study a neurological disease. How can doing these perturbations in the genome help me? I think we need to look broader and ask, how does the genome function, and look at how do we encode information? And people have said it's too difficult, but I would argue that with these technologies we've got, it might be possible. And in addition to that, we're talking about, like within regulatory elements, perturbing them and seeing what happens. And we know that there's a lot of redundancy. For example, what types of enhancers are redundant, what types aren't? Can we see patterns in that? And I think those sorts of general questions could be informative to many people.
I think this is a very important point that you're making, but I would like to get back a little bit at you, because I think that it's a very difficult problem to solve if you try to solve it in general. But there are exquisite examples, for instance, in how superenhancer may be collapsed by targeting just one single element within the superenhancer, which is a 15 kb region. And many, many studies have been done to understand the role in enhancer in promoting specific phenotypes. So I think that if we limit ourselves and we are a little bit less grandiose in understanding everything, there are lessons that could be learned that can then be generalized to learn across many other phenotypes. So I think that what you're saying is that the technologies are there, completely agree. I think it would be difficult to use them even today, even 5 years from now, to understand how enhancers work in absolute terms.
I don't mean that. I mean, for example, you have you have highly conserved enhancers, you have enhancers that are not very well conserved. If you were to silence them and see, well, what's the difference between them? Or even all of these putative enhancers we have, you could build reporter assays to test, do they drive expression in the cell type that you expect? I feel like there's the aspect of how do we get at disease and how do we understand true phenotype. There's also how do we use these to understand how sequencing codes function.
So we are completely out of time, and so at this point I think this was an extraordinarily successful session. Thanks to Len and Barbara, and thank you.